So, hello everyone, myself Praneet, and today I'm very happy to share that I have just added a new module to CSBB-Shiny. So CSBB stands for Computational Suite for Bioinformaticians and Biologists, and this is a Shiny interface for CSBB. And today I added a new module called Single Cell RNA-Seq Analysis. So now you can basically do a quick single cell analysis for your transcriptomic data. And let me quickly give you a get overview of how does it work, what are the functions, what are the things you can do with it, and then we can take it from there. So as some of you might have already seen this and might have already used it for some of the analysis, now CSBB is packed with 8 modules: visualization, normalization, basic stats, differential expression, correlation profiles, function, pathway enrichment, chip ATAC analysis, and then finally single cell RNA-seq analysis. And just in case if you have not seen a tutorial before, there is a video tutorial which you can get it from here. It's here on YouTube, and then you can watch it to how to use the other modules apart from the single cell. So today, in this tutorial, I will be covering how to use the single cell module. So just to give you a quick overview, this is available on GitHub. Whole code is on GitHub, and you are welcome to give your suggestions, improvements, issues that you face. And also you can use it on your own servers by deploying it on your own machines. Now, without wasting any further time, what we'll do is, let me quickly go to the result window because the two main components of CSBB-Shiny is the result window and the visualization window. So visualization window basically will show you plots, whatever is derived from the analysis, and the result window will show you the markers, or maybe let's say some information that you can get from the single cell analysis here. So when I go here, you will see there are all these modules that I just listed out. So we'll go ahead and select the single cell RNA-seq analysis. Now there are, the first thing that you see, it's select data type, input type. And right now it provides you three options: raw counts matrix, H5, and R object. So raw counts matrix is just a counts matrix of genes, and then their counts across cells, which you can input. The other one is H5, which basically if you put here, it will ask you to give it HDF5 file or H5 file, which comes out from Cell Ranger. And the last one is R objects. For example, you have already processed your data through Surat, and you want to now quickly generate some plots and stuff. You can just upload your processed R object here. Now, give you a little brief information of what is the engine behind CSBB single cell module. So I have used for now Surat, because Surat is one of the most widely used, and I would also say it's most cited analysis toolkit, which people across the world are using to analyze single cell data sets. And the main reason for creating this module is just to give a more feasible options to people who don't want to code or don't want to use R command line or console versions, and they can just come to this interactive web page where they can input their data, play with the parameters, and generate different plots, analysis, and use that for their research or grants or whatever purposes they're using it for. So let's say today I'll give you a demo of both raw counts matrix and a H5 matrix, H5 file. First, we'll start with raw counts matrix. So it comes with an option of you can upload a raw counts matrix. Then there's an option of selecting species, whether it's human or mouse. And I would say that in coming weeks, or maybe I think as soon as possible, I'll be adding more species to this interface. Then there is a box where you can input the number of variable features you want to use for the analysis. Now, by default, it's set to 2000, but you're welcome to play with it. Then same with the number of dimensions. This is the number of principal components that you want to use, which is again set to 10. Resolution for clustering is set to 0.5, and that is, I think, these are all the default parameters you'll see on the Surat vignette for clustering analysis of single cell data. Then the other one is select dimension reduction, where you can select either UMAP or Disney. UMAP is uniform uniform manifold approximation projection algorithm, and Disney T stochastic neighbor embedding. And then finally, there's another way, like there are some ways to visualize your data where we have different options. The first is gene expression plot, which is in simple terms, you might be aware of feature plot. Then dimension reduction plot, where you can take a ask CSBB to draw Disney or UMAP plot. Then top 10 markers heat map, and violin plot and dot plot. And all of this I will be covering in this tutorial today. And finally, there is a select genes, which will start showing you genes once you start uploading your, like once you upload your matrix, and then you can just add genes for feature plots, volume plot, dot plot, and so on. So without wasting any further time, I'll go ahead and upload a file here. I have a raw codes file. I think it has some 20,000 genes and 1,000 or so cells. And once the upload is complete, you will see that this has kind of written down. Now, what happens is it will take some time to upload the data to the server and then process it in the background. And once the data is processed, it will directly show you a marker file for all the clusters it has found. So with the current parameters set here, like 2000 variable features, 10 dimensions, 0.5 as resolution for clustering, I think there were 4 or 5 clusters that I saw, and should be up in a second. It takes some time, so what we have to do is kind of sit tight and wait for it to kind of project everything on the server or on the interface. And in the meantime, what you can do is you can even go to visualization window. So see, as I selected visualization, so the UMAP plot was ready. But before going to that, I'll first go to result window. And what you can see is there is a table which shows you the markers for the clusters. So there are 59 pages, but what you can do is you can go here and change it to, let's say, 50 entries. So you'll start seeing 50 markers per clusters, and then you will have 0, 1, and so whatever clusters you have. The other feature that I really like is you can go ahead and search for a gene of interest. For example, let's say I want to search for hex. And as you do that, you'll see hex and which cluster it was found in, what was the p-value, what was the log fold chain, and so on. Then you can download this using a button called download results. So I'll go ahead and move it back to 10 entries. So it's better to see on the screen. And then once you hit the download results, it will download the whole markers file to your system. Then once I go to the visualization window, what you can see here is, this is the UMAP plot, which basically shows you how these different clusters or cells are clustering. And there are 5 clusters that we see. And now let's say I want to do a gene expression plot. So right now it gives an error saying that there were no features selected, which is true because we have to still select the genes from here. So let's say I will randomly select certain genes, and I'll just keep selecting them, maybe let's say 4 or 5 genes. So you can clearly see that there are 6 genes selected, and it has created a feature plot for all the 6 genes at one time. I have tested it for 10 or 12 genes. It works fine, but you're welcome to try it more and maybe even add to the code base. Then as you see that I have randomly selected these genes, so you can see there's only one gene that is kind of ubiquitously expressed, which is NDUFS1. Then I can add one gene which I know, for example, FOXA1, which is again tells you how easy it is to search for a gene and then add it to the list. So let's say I go ahead and add FOXA1. Now you'll see that FOXA1 is now added to the screen. And the best part is you can go ahead, select the save plot option, and the results or the plot will be downloaded to your system. Now let's say the other cool feature that I think is present here, is that you can go ahead and do the same for violin plot. Now, once you do the violin plot, the best thing is that this list doesn't change. You can go ahead and definitely change it, but the best part, for example, let's say you are looking at the same genes, but you're looking in different formats like feature plot, violin plot, dot plot, and so on. So this is a very easy way to do this. Like you can set the gene list, like the same gene list will be appearing there, and then you can change the plots from this option. And then let's say I want to do a dot plot, which is again a very cool feature in Suite where you can see how these variable different markers are expressed across clusters, and they could be easy way for the eye is to interpret what are the markers for certain clusters and so on. The other feature that is here is top 10 markers heat map, and as you click on that, what happens, it will show you the top 10 markers for each cluster that it has found and display the heat map for that. Now again, it's very cool feature because what you can do is you can quickly, first of all, download the plot, and then you can also see quickly what are the top markers. Like instead of going through the huge list of markers and finding out the top ones, you can just get a heat map and see which is the top markers here. Now, after you have done this analysis, you have played with it. Let's say you went ahead and changed certain features and your results will change accordingly. The other part that I think is very important here is that you can download that Surat object, right? And the reason behind that is because once you have downloaded the object, you'll have everything stored in that object, and you can maybe do custom analysis on your own laptop, or as I said, there's another feature as that there is a feature of again uploading the same object, and you can upload that object and create more plots and visualization, or maybe just redo some clustering and stuff. Now, one thing to remember is that by default, CSBB saves the name of the object as surat.object, and it has been mentioned in the getting started page as well. But if you're uploading your own Surat object, it's recommended to set the name to surat.object. Then let's say I want to process a H5 file which directly is outputted from Cell Ranger when you are analyzing single cell data. So what I'll do is I'll go ahead and just quickly refresh it so that the memory is refreshed. And then I'll go ahead and select single cell RNA-Seq. And then here I will do H5. And again, I'll go to the result window. And I think this data has 10,000 plus cells. So definitely it'll take more time to process it. But again, in the meantime, I can show you other pages and then we can wait for the results to pop up. And let's say I browse here and get a H5 file from my system, which is right here, and load it. So once it has uploaded, you can go ahead and let's say, I can see on my screen that it's doing all this things where it's scaling the matrix, normalizing it and stuff. And I can go to the get started page where I have kind of mentioned everything that I was talking about. One thing that I wanted to specifically mention is that CSBB does a quality filtering on your cells, where I have by default set it to 95th quantile. So the two main things or two main parameters that ones look for when kind of removing cells which are not important in the analysis is percent of mitochondrial expression and number of features that are expressed per cell. So by default, what I've done is it's set to 95th quantile for each. So whenever there are cells which are expressing, for example, let's say there are 1,000 cells, and then most of the cells are expressing 5,000 genes, right? But there are a few cells which are expressing just 10,000 genes, which are way above the cutoff. So what I've done is kind of set a very default or a very minimal threshold of 95% quantile on both to remove cells which are expressing just way beyond what is expected out of, or which is seen from all the other cells in the analysis. Now, one thing that I want to share here is that for the first preliminary analysis or the first core, you should be using very minimal cutoffs or less stringent cutoffs so that you can see the overall data, and then you can go ahead and dig in and then remove more cells from it. This is one thing. And then if I go back to the result window, and I think the result should be popping soon because it's doing the marker analysis for the 13th cluster. Some more advancements that would come to this package soon enough would be, first of all, removing cell cycle, which is very easy to add. And the thing is, it should be rolling out very soon. And the other thing is giving you different options of normalizing the data. So by default, it uses log normalization, which is Surat's default normalization. But I'm planning to add SC transform, which is again a great feature that Surat has, where you can further kind of normalize your data and clean it up, remove effects or batch effects from the data, and then show it or even compare it. So that's something in the works, and that should be rolling out pretty soon. As you have seen, and then I wanted to mention again that it just takes some time to reflect everything on the screen. It's because it's processing and loading everything, packaging everything up, and then putting on the screen. Once it has done everything, it's very quick in creating plots and stuff. But yeah, you have to bear with it because it's running on 8 gigs of RAM, so it doesn't have that much power right now. But if you're hosting it on your own server, or let's say your own machine where you have tons of RAM to use, I'm pretty sure it would be much quicker and faster in response. So the other thing, as I said, when I was showing the previous data, like whenever I switch to a visualization window, the results pop up. Anyways, so again, I will go back to the result window, and again, you can see that the first cluster 0, which is, it seems to be it's a mitochondrial cluster because all the mitochondrial genes are kind of very high here, right? But you can also again go ahead and search for your genes. For example, let's say I want to see IL-1 or let's say related genes. I can see what clusters they are in, and then accordingly track my clusters or annotate them. Again, going back to the visualization window, so you can see here a beautiful UMAP plot where you have these clusters and then how they're connected on a UMAP plot. Now, UMAP has one good thing that I have kind of read up different places, that you can see the global structure of the cells. So for example, if you're seeing this 3 is very far away from 7, that means they're transcriptionally very different. That is one thing that you can make out even from the UMAP plot, which you can't do it from the TISNE, because TISNE doesn't preserve the global structure, it preserves the local structure of the cells. Then again, for example, let's say I want to do a gene expression plot. It will automatically have all the genes listed here from your object. Sometimes you might see a problem. For example, let's say I click this one, this was present, but there might be just certain genes which it will show or say that was not found. And but the whole reason for that is that it was just discarded from the analysis when Surat was kind of filtering the cells or normalizing the data, right? So that's something that could happen, or maybe the gene was not even expressed, it was just thrown out. And then again, I'll just keep adding some random genes here just to show you or give you an idea of how does the overall analysis looks. So for example, again, I by default or by randomly just select certain genes. Most of them are not expressed, but there are a few which are kind of expressed at certain clusters. Again, you can download the object and you're welcome to play with it. And one thing that before I wind up the tutorial is that once you go here and you say, let's say, go to the R object, you'll see one thing, that these options are kind of limited here now, because it will take your processed object and then basically you can select species, you can create all these visualization plots, but there would be a few more analysis that would be coming in. For example, you can redo the marker analysis, you can, let's say, redo the clustering using UMAP if you have just had the TISNE before, or let's say set the resolution down, up, and further, all these things. Which now, for example, and I forgot to mention, for example, when you were back here and you were working on this analysis, so when you switch back and forth, what happens is, and in terms of the parameters, so for example, let's say you change the resolution clustering to 0.5, right? And then you added 0.6, or let's say you went to 1. All these will be stored in the object which is being processed at the background. So when you will get the final result, you'll have everything in there. So that's another cool feature where, for example, you have tried multiple things, you have everything ready with you, and then you can just churn it out. We go back to this app or application, and you can create new plots, or maybe just look at the data yourself using more custom scripts analysis. So for now, this is all from my side, and I'm very happy to say that this is something which I was kind of hoping to have launched for some time, and now it's kind of becoming a reality. And I think with time, I would be able to add, and the plan is to add more analysis toolkits, like Surat is one, but there are others like SCRAN, and then there's Spring, and so on, which could be easily, I think, integrated into this framework, where people will have more flexibility of using the toolkit they want to use, rather than just using Surat here. But from my experience, I have like worked on a lot of different single cell analysis or single cell data sets, and so it just works great. I think it's very easy to use, very easy to understand, and yeah. So that's why that was one of the reasons that why I added for here for the first time or the first go. But thank you again for your time and patience. And if you have any questions when you're using this, please do go to the GitHub and give your issues, feedback, comments. And if another thing that I wanted to mention before I end the tutorial is, I'm very open to the collaborative code base. So if you want to contribute to the code base, make it more fancy, better, I'm all welcome. I'm all welcoming for it. So do get in touch if you want to contribute to the code base, or you want to kind of do a pull request to certain elements. I would love to hear from you. Thank you again for your time and patience, and I hope CSBB helps in your analysis, research, or your just one kind of learning things. Thank you. Take care.